now that we're done with glycolysis, let's continue to pyruvate dehydrogenase complex. So we know glycolysis happens in the cytosol of the cell, and glycolysis gives us pyruvate, which is the end product. So once pyruvate is synthesized, it'll hop onto a protein called pyruvate mitochondrial carrier and then make its way to the inside of the mitochondria. Now, before we continue, let's clarify what pyruvate dehydrogenase complex is and is not. First off, it is not part of the Krebs cycle. It is, however, an intermediary step that connects glycolysis to the Krebs cycle. And the goal is to take pyruvate, and this right here, pyruvate, and convert it to acetyl-CoA. So there are two things that we should master for talking about pyruvate dehydrogenase complex, and that is the structure of pyruvate and structure of pyruvate dehydrogenase complex. So pyruvate is made of 3 different carbons. The first one is attached to a carboxyl group, this one right here. The second one is attached to a carbonyl group. And lastly, we have the methyl group. Now, moving on to the structure of the complex. So this complex is made up of 3 different enzymes. This is the first one, second one, and third one. So the first enzyme is called pyruvate dehydrogenase, also known as E1. The second one is called dihydrolipoamide acetyltransferase, also known as E2. And lastly, we have dihydrolipoamide dehydrogenase, also known as E3. So each of these enzymes have a cofactor. For E1, it has thiamine pyrophosphate, or also known as TPP. And this comes from vitamin B1. And next we have, for E2, it has 2 different coenzymes, lipoic acid and coenzyme A. The coenzyme A comes from vitamin B5. And lastly, we have cofactors for E3. And these are FAD, which comes from vitamin B2, and NAD, which comes from vitamin B3. Okay, so now that we're familiar with this, let's move on to the actual process. So when the pyruvate comes in, it attaches to this cofactor, which was called TPP. And as soon as it happens, it releases a carbon, and now it goes from pyruvate to hydroxyethyl TPP. Next, the TPP then transfers it to E2, and here it attaches to lipoic acid, and specifically this is known as the thiol group. The thiol, it attaches to the thiol group, and then pay attention to the TPP. It goes back to its original place, and now it's ready to take in more pyruvate molecules. So coming back to E2 now, we have acetyl-lipoamide. The lipoic acid here does a little loop-de-loop and tosses it away to coenzyme A. And finally, we have acetyl-CoA. This can now leave the complex and make its way to Krebs cycle. So now let's come back to lipoic acid. So as a parting gift, when the acetyl group is detached from lipoic acid and attaches to acetyl-CoA, it leaves a parting gift to lipoic acid, and that's these 2 atoms right here. These are hydrogen atoms. So now it's known as dihydrolipoic acid. See, the problem now is in this form, it cannot accept any more molecules, and it has to be converted back to lipoic acid. So that's where E3 comes in. We have FAD right here. So FAD will accept these 2 hydrogen atoms, and it's now converted to FADH2. Okay, now what about FADH2? Well, luckily we have NAD plus that comes in. So it kicks one of the hydrogen atoms out, and that's turned into proton. and then the other one attaches to the NAD and turns it to NADH. And now we know that NADH can make its way to electron transport chain and synthesize ATP molecules. Now, moving on to regulation. So the complex, the pyruvate dehydrogenase complex, is regulated by two main enzymes, PDH kinase, which will inhibit E1 activity. And then on the other hand, we have PDH phosphate. This will activate E1 activity. Now we have these three different molecules and they activate PDH kinase. Take a look at these molecules. What do these all have in common? They all signify to the cell that there is enough energy, right? So if there's enough energy, they can turn on the PDH kinase, which will then inhibit E1, stopping the conversion of pyruvate to acetyl-CoA. Now, we have pyruvate on the other hand, which will inhibit PDH kinase. So once pyruvate turns off the kinase, now we have nothing that stops E1 from functioning. And lastly, we have calcium. Calcium activates PDH phosphate, which will then in response activate E1, increasing the activity of converting pyruvate to acetyl-CoA. Moving on to clinical correlation. So first we have E1 alpha subunit mutation. This is an X-linked disease, and just like the name says, this is alpha subunit mutation. So what exactly is alpha subunit? So E1 is made up of 4 different subunits, 2 alpha and 2 beta. Just like the name says, the alphas in the E1 are mutated. So we'll usually see infants come in with lactic acidosis, and they also have developmental delay, and sometimes, unfortunately, it can lead to early death. Moving on to arsenic poisoning. In arsenic poisoning, we'll have arsenic that will come and attach itself right here, and it attaches to the dihydrolipoic acid form. And now we know once it attaches to this group right here, there's no going back to lipoic acid, and this will end up halting the pyruvate dehydrogenase complex, meaning no acetyl-CoA production, eventually leading to less ATP production. So depending on if it's acute or chronic, we'll have different sets of clinical presentation. So in acute, we'll have garlic breath, multi-organ failure, and GI distress, including vomiting, diarrhea, and abdominal pain. And in chronic, we end up with peripheral neuropathy, changes in skin pigmentation, and cancer. And that's it for today's lecture. If you enjoyed it and if you learned something, make sure to give it a thumbs up and don't forget to share with someone who might be struggling in this topic.